Hello, my name is Mackenzie Venom, and today I will be talking about venom physiology. Because venom is such a very broad topic, this will be more of an overview of venom physiology, what venom is and what effects it has, as well as its possible uses in therapy. And I will mostly be covering snake venom since that is very highly studied, but there will be some other types of venom mentioned as well. So I'd like to start by just defining venom. Venom is, in the broadest sense possible, a range of proteins with varying effects on the body. And these normally contain varying amounts of enzymes such as phospholipase A2, which cleaves phospholipids and therefore, of course, has a great effect on vesicles and the plasma membrane and any phospholipid bilayer. Three-finger toxins, which are non-enzymatic, they are not enzymes. And these are both nicotinic and muscarinic acetylcholine receptor inhibitors, which of course means that they will affect any sort of skeletal muscular activity by prohibiting those action potentials and limiting movement in skeletal muscles. And then metalloproteinase, which is kind of exactly what it sounds like. It is a proteolytic enzyme with a metal atom attached to it, which is normally zinc. Hyaluronidase, which is difficult to pronounce, promotes higher permeability in membranes, which causes leakage and therefore causes inflammation and quite a lot of pain. And acetylcholinesterase, ACHE, which you've talked about in class, which degrades acetylcholine in the synaptic cleft and also inhibits action potentials and skeletal muscular movement. And all of these have different functions. They can be myotoxic, which means that they have an effect on muscle tissue. They destroy muscle tissue. Cardiotoxic, which means that they inhibit heart function. Hemotoxic, which means that they destroy tissue in general and also have an adverse effect on the bloodstream and blood cells. And, of course, neurotoxins, which means that they affect the central and peripheral nervous systems and neurons in general. As you can see from the figure on the side there, that kind of illustrates the effect that venom can have on proteins. It destroys tissue, it causes bleeding, and generally harms tissue, of course. And many of these also inhibit nicotinic acetylcholine receptors, as previously discussed, destroy vesicles, or degrade acetylcholine in the synaptic cleft, which makes it very relevant to talk about after the class has covered muscular functions and the nervous system. Apologize, we're not... There we go. Sorry, technical difficulties. Having to record this at home. And to further illustrate the different elements of venom, we have this figure right here, which illustrates the different components of snake venom. Snake venom is made up of protein and peptide components, and also a collection of non-protein and peptide components. These could be just free amino acids, lipids, carbohydrates, metal, things like that. And within the proteins and peptides components, we have enzymatic properties and non-enzymatic proteins, basically things that are enzymes and things that are not enzymes. And as previously mentioned, we have proteolytic enzymes, which disassemble protein strands. And these are found in very high concentrations in hemotoxic venom, which is primarily found in the Viperidae snake family. This includes vipers such as pit vipers and also rattlesnakes. And if you've ever seen a rattlesnake bite, it causes tissue necrosis and also heavy internal bleeding. And to illustrate that more, we have SVSPs in the enzymatic properties section there. These are serine proteinases, and they affect platelet aggregation, which means that they cause heavy internal bleeding that doesn't immediately clot. The wound doesn't close up easily because platelets cannot aggregate to close up the wound, and therefore there's just a lot of internal bleeding. And then, as previously discussed, ACHE breaks down acetylcholine in the synaptic cleft and reduces motor end plate activation and therefore any sort of skeletal muscle movement. And then, of course, we have a lot of non-enzymatic properties that act on receptors and ion channels and further inhibit movement by being unable to release calcium and sodium ions. And from here, I'd like to talk about how certain animals get around the effects of certain types of venom. The first animal I'd like to talk about is the pallid bat. And pallid bats are native to North America, and they are resistant to Arizona bark scorpion venom, which causes pain. Pallid bats forage on the ground, unlike other bats, and of course that means that they are near these bark scorpions, and that makes stings very likely. Now, normally, bark scorpion venom is quite potent. It is incredibly painful, and it occasionally kills humans. It is the most potent scorpion venom in the United States. And it targets specific binding regions of voltage-gated sodium ion channels. And in the pallid bat, there are 2 amino acid differences in that binding region that prevent activation of the channels by this scorpion venom. And this difference is also present in Virginia opossums and Tasmanian devils, both interestingly non-placental mammals. Researchers believe that this could be convergent evolution, but we aren't exactly sure. There isn't a whole lot of study on the evolution of that yet. And another a bit more famous example of an animal that is immune to a certain type of venom, the same type of venom actually, bark scorpion venom, is the southern grasshopper mouse. These are also found in Arizona. They have a similar adaptation to the pallid bat in that there is an amino acid sequence variant that causes the venom toxic to bind to a non-target channel, and it avoids the action potential propagation, which means that the pain signal is not propagated. What's interesting about this is that the venom actually dulls the pain signal entirely. And this is interesting because pain resistance adaptations are fairly uncommon in general because, of course, pain is a very important thing to experience. It indicates that there's damage being done to tissue and that something needs to be done to stop that damage and respond before it permanently harms the creature. Something that I thought was worth noting is that the researchers that did the original study that was published in Science about this adaptation in grasshopper mice believe that there may be an evolutionary arms race between the bark scorpion and mouse, in that the bark scorpion venom is constantly changing over time in composition, and in turn the grasshopper mice have to respond to this, or else they will lose their immunity to this venom in this scorpion that they rely on for food. And now finally I'd like to talk about how venom proteins can be used for therapy. Obviously, because venoms are such a broad topic to study, there is a huge library of proteins to examine that could all be potentially used to treat different diseases. For example, we have proteolytic enzymes that interfere with platelet assembly and blood clotting, as we mentioned a couple slides ago. And this could actually be used to treat heart disease and thrombosis because, of course, it prevents blood clotting and blood pooling. And then other venoms, such as rattlesnake venom, cause very fast coagulation of blood, and these quickly stop bleeding, which could be useful in surgeries. I'd also like to illustrate that snake venom has antimicrobial properties. The proteolytic enzymes, of course, can destroy harmful bacteria, and this may be very important as antibiotic resistance increases and traditional antibiotics become less effective. Something I thought was very interesting to highlight is that snake venom can also interrupt the life cycle of viruses such as HIV and hepatitis C, which means that these could potentially be used as therapy in the future for people who would otherwise have incredibly just crippling diseases to live with. And of course, just due to the ability to disassemble protein structure, all of these different types of venom and all of their proteolytic enzymes could potentially be weaponized to fight multiple types of pathogens. Thank you very much. I hope you found this as interesting as I did. I apologize for the format, but I'm doing my best to film this at home as well as I can. Thank you very much.